So question number 4, they are asking lab diagnosis of intestinal amoebasis, not amoeblasis. Okay, so lab diagnosis of intestinal amoebasis, that means he is talking about Entamoeba histolytica. Okay, so here as you can see, what is this and what is this? This is the trophozoite form of the Entamoeba histolytica. What is Entamoeba histolytica? That is a parasite. What kind of parasite? Protozoan. So it is a protozoan parasite, single celled. So this form, this huge comma shaped thing, this is nothing but the Entamoeba histolytica, which is in the trophozoite form, and this is the cyst form. Now if you ask what is the meaning of trophozoite, trophozoite is nothing but it is the active motile feeding stage of the protozoan organism. Okay, maybe Entamoeba histolytica or maybe Giardia lamblia. So this is the active motile feeding stage. Then if you ask what is the meaning of cyst, cyst is nothing but like a protective resistant stage. So when the environment is not suitable, this trophozoite will convert into cyst stage so that it becomes dormant. So this is the trophozoite form, which is the active feeding stage, and this is the dormant stage, that is the cyst stage. So this looks like this and this looks like this. So this is the matured one, the quadrinucleated cyst. Quadri means 4. You can see 1, 2, 3, 4. 4 nucleus. And this will be given in microbiology lab exams. Okay, they might keep a picture of this, or they might put the slide under the microscope, and you have to see this and understand whether it is what kind of cyst. Like they will give Entamoeba coli also, then they will give Entamoeba histolytica. In Entamoeba histolytica we have 4 nucleus. Okay, see, but in Entamoeba coli we have more than like 6-8 nucleus. It looks similarly, but they have 6 to 8, and that will be much bigger than this. Okay, that is Entamoeba coli. So do not get confused in the practical exams. So we understood the basic things. So we have the trophozoite and we have the cysts. Okay, next we will see the life cycle. In the life cycle, what do we have? So first, the infective form. The infective form means what is going inside the body. Here the cyst usually goes inside the body. Infective form is the cyst. Okay, the quadrinucleated cyst. It enters into the stomach from the mouth. Okay, buccal cavity. So yeah, in quadrinucleated cyst, it is entering into the stomach. Then here we have excistation. What does it mean by excistation? Excistation means the cyst is slowly converting into its active form. How? So there are 4 nuclei. As you can see, the cyst is broken here. The things inside the cyst are coming out. Then there is a multiplication kind of stage, okay, called as metacyst, and then this is getting converted into metacystic trophozoite. So what is trophozoite? Trophozoite is nothing but the active feeding stage. Now this trophozoite is slowly developing. See here, this is the trophozoite with a single nucleus. So multiple trophozoites will develop, and this is where, this is the small intestinal part. Then they are entering into the large intestine. So here as you can see, this is slowly growing, and what is this? Intestinal invasion followed by direct and hematogenous spread. So they are invading the intestine, also they are spreading through the hematogenous, that means blood, or direct also. Okay, direct from the intestine into the other areas, adjacent areas. Okay, then they are passing through the large intestine, the transverse colon, and then they are entering into the left side of the colon. Okay, so here we have the encystation. What does it mean by encystation? Excistation is when this cyst was bursting out to develop the trophozoite, and now this trophozoite will convert again into the cyst. Okay, so trophozoite is converting into precyst, then metacyst, which has the uninucleate, then the binucleate, and then the tetranucleate, and then this comes out. Okay, this is also the diagnostic form. Diagnostic form is both. Trophozoite also can be found, cyst also can be found. Okay, so it's seen in the active infection. Trophozoite is seen when there is an active infection, and cyst in feces, this is found when it is in the carrier state. Okay, So did we understand the life cycle? See, this is a quick revision video. Okay, they are asking just for the 3 markers. See, they are asking for the 3 markers. That also they are asking the lab diagnosis. What am I discussing? I am discussing from the basics as a quick revision, so that you don't feel lost. And we will discuss the lab diagnosis also, but all these topics, they need a dedicated video for themselves. Okay, so we will make a dedicated video about this everything again in details. I'm just quickly rushing through the points, okay, so that I can also revise. And these are the complications. Like we have the flask, the famous flask shaped ulcer. This is where this is in the intestinal wall. Okay, so flask shaped ulcer, see here. Then there is formation of this huge sinus, deep extension of the ulcer beyond the muscularis mucosa. Then they are causing perforations. Then there is, see here, there is the amoeba. Then there is invasion of the blood vessels. So these are the complications. Then there is this famous complication of the Entamoeba histolytica, that is apart from the intestinal. This is the extraintestinal, the famous amoebic liver abscess. Okay, so this is done. Next we will see the lab diagnosis. This is the thing that should be discussed in this video, but anyways, so lab diagnosis, we have the stool microscopy by wet mount permanent stains, etc., that detects cyst and trophocytes. So what was coming out of the body? The trophocytes or the cyst. Trophocytes in case of the active infection, cyst in case of the carrier infection. So both can be detected, depends on which stage he is. So what are we testing here? Are we testing saliva? No. Are we testing tears? No. What are we testing here? We are testing the stool. Then we can do the stool culture, inside which we have the 2 things. One is polygenic culture, the other one is anexenic culture. So here we have that diamonds media and all. Okay, here we have that National Institute of Health or something. Then we are checking for the antigens, also called as copper antigen present in the stool. So these all things are used in the detection of the antigens. Then we have the serology, amoebic antigen by ELISA, amoebic antibody by all these things. Isoenzymes, zymodine analysis, molecular diagnosis, nested multiplex PCR and real-time PCR. We will go in detail. So laboratory diagnosis, minimum of 3 stool samples on consecutive days as amoeba are shed intermittently. Okay, so we need minimum of 3 stool samples. Okay, then stool microscopy. So you will take this stool and you will check under the microscope to demonstrate whether there is presence of trophocytes or quadrinucleated cyst. So trophocytes indicates active infection, quadrinucleated cyst indicates the carrier infection. Cyst and trophocytes of Entamoeba histolytica should be differentiated from E. coli, which is a common cell in the stool. So Entamoeba coli. Okay, this Entamoeba coli is the common cell. I have told already Entamoeba histolytica, they have, if they are searching for this cyst, if they find this cyst, then there will be like 4 nucleus in the cyst. But in Entamoeba coli, there will be more than 6 and 8 nucleus in the cyst, and they will be huge. I mean, that will be larger in size. Okay, what is the meaning of polygenic culture or axenic culture? See, polygenic means the culture media, they have more than 1 organisms. Okay, maybe along with this thing, what is this amoeba, there is other bacteria. Okay, and why is it important? It is important because so that it mimics natural environment for the organism that we need to develop. Okay, so maybe in this case the amoeba, they grow well along with the presence of the other organisms. Like here they contain the bacterial supplements, okay, which provides the nourishment to amoeba. Hence it is called as polygenic, means multiple other organisms are also present along with the target organism that we need. Okay, what about axenic? So in this case it is not there, so other organisms are not there, only this organism is there. Somehow they are developing these single organisms here. Okay, so that is the difference or the meaning of the polygenic culture. So polygenic means they are trying to mimic the natural environment so that the target organism grows well. Okay, then used in the chronic and asymptomatic carriers passing less number of cysts. Okay, so if a person is passing less number of cysts, then we are using this polygenic culture. 50 to 70% sensitivity and 100% specificity, gold standard. So polygenic culture is the gold standard. The various culture media used are National Institute of Health media, Boyk and what is this? Dr. Boholav or Darboholav. X serum medium contains Loki's medium or Locke's solution. Then we have Balamut's medium and Nelson's medium, Robinson's medium. Okay, too many mediums. So polygenic culture, why it is used? It is used for the chronic if there is less number of cysts. Then we have the axenic culture. It lacks bacterial supplements. Example, Diamond's medium. Diamond's medium. Why is it called Diamond's medium? Not sure. Are there diamonds inside? Really? Axenic culture is useful when the bacterial flora interferes with the test results, such as. So why is it useful? When the bacterial flora interferes with the test results, such as studying pathogenicity of the amoeba, testing anti-amoebic drug susceptibility, preparation of amoebic antigen in mass for serological test, for harvesting the parasite to determine the zymodine pattern. Then stool antigen detection, copper antigen. So we're detecting everything. Like in microbiology, if you do not write ELISA, you are not a microbiological student. Okay, you have to write ELISA for everything. That is mandatory. That is the culture. That is the pratha. Okay. So detecting lectin antigens, amoebic antigen in serum indicates recent and active infection. ELISA is done for lectin antigens. So amoebic antigen, lectin antigen. Amoebic antibody, serum antibodies appears only in the later stage of the intestinal amoebiasis. Example, ELISA, IFA, indirect hemagglutination test. We have zymodine analysis, detection of the isoenzyme markers like malic enzyme, hexokinase, isomerase. Nested multiplex PCR, Charcot-Leyden crystals in stool and moderate leukocytosis. Charcot-Leyden crystals are needle-like structures formed from the breakdown of the eosinophils, a type of breakdown products of the eosinophils. Done.